Our next one is going to be EMB Agar. Now, EMB Agar, once again, is going to be a gram-negative type agar. It's going to be used for gram-negative species. You put a gram stain, you'll see it underneath the microscope. It comes back gram-negative, usually going to be your pink gram-negatives, and you're going to stick them on a McConkey plate or an EMB plate, and this will help you figure out what you're looking at. Now, this particular agar is differential and selective. So it is selective, and let's talk about what does the killing in this. Now, in this case, EMB agar stands for eosin methylene blue. Eosin and methylene blue. Eosin is a dye, which gives it this purple color. Methylene blue is also a dye, the same thing that we use for simple stain. So eosin and methylene blue are dyes that have been added to our nutrient agar, essentially. Eosin and methylene blue, both of those dyes do really well together to kill most gram-positive species. But most gram-negatives can survive in the presence of those two dyes. Most gram-positives can't. So that's what our selective agent here is, the presence or absence of those dyes. Can you survive in the presence of them or not? Now, the differential agent in this case has to do with what these bacteria are going to do with the presence of those dyes. Now, this is kind of interestingly weird. This agar contains lactose, and technically that's the differential agent is lactose. It works on the same concept. Can you eat lactose or not? If you can eat lactose, you produce a basic an acidic byproduct. If you can't eat lactose, you eat the proteins, producing a basic byproduct. Now, in this case, there is no pH indicator. So how are you going to tell if you can make acids or bases? Kind of interesting. So if you can eat lactose, you are going to ferment your lactose. You're going to produce an acid. That acid is going to be released outside of the cells, which will then diffuse into the agar. Now, in this case, acid, what it does when it comes in contact with the bacterial cell walls, and this was regardless of what type of agar it is. The other agars just don't have the types of dye that will work this way inside of them, or else they would do this, but they don't. Well, anyway, acid comes in contact with bacterial cell walls and cell membranes and things, and it kind of makes them a little leaky. And what that does is it causes the bacteria to suck up the dyes from the media. They suck up different types of dye. Whichever type of dye you suck up tells us what type of sugar you are fermenting. Whichever type of dye you are sucking up tells us if you can ferment lactose at all, if you suck up a dye in the first place. And then whichever type of dye you suck up allows us to tell apart the different species, and that's pretty neat. Now, this particular agar is specifically really only designed for E. coli. It's used mostly in the food and dairy industry for a very quick test to see if E. coli is present in your food. So, what's going to happen here? If you can grow on this particular plate, you have been able to survive the selective agent, which in this case is eosin and methylene blue, which means you're a gram-negative organism. You survive the selective agent. Now, the differential agent is lactose. You are able to eat lactose. You eat lactose, you produce an acidic waste byproduct. E. coli. E. coli is our champion here. This is E. coli right here as well as right here. E. coli produces an acidic waste byproduct. It can eat lactose. It starts producing that acidic waste byproduct. It's released into the environment, into the agar, and it's going to cause the E. coli cell to suck up large amounts of methylene blue from the environment. That methylene blue gives the E. coli that green shimmery tint to them. And that is characteristic of E. coli. They are the only species that can do this for our purposes. And if you see that green shimmer, you have E. coli. No matter what else grows around it or on it, and if you see the green shimmer, it is E. coli. And this is why dairy industries and food industries use it, because it's so easy to tell apart E. coli. All the rest of the gram negatives, if you can ferment the lactose, will uptake eosin. And eosin gives them this purple color, either kind of a pinkish version, depending on how much eosin they uptake, or a really dark purple. It just kind of depends. So this is a really dark purple version, or kind of a pinkish bubblegum color. So everything other than E. coli, every other Enterobacter ACE species, Enterobacter is going to uptake eosin, which gives them that purple color. Methylene blue for our E. coli, and that's the key here. That acidic-based byproduct makes their cell walls leaky. E. coli sucks up methylene blue. Everybody else sucks up eosin. Now, what happens if you can't eat lactose? Well, if you're somebody like Salmonella who can't eat lactose, you're going to eat the proteins in this agar, the default answer. You're going to eat the proteins. Those proteins are going to be hydrolyzed into a basic byproduct. That basic byproduct will be released out of your cells into the agar. And bases do the exact opposite of acids when it comes to the cell wall. They make the cell walls condense. They make the cell walls harder. So basic pHs make cell walls dense. So poor little guys like Salmonella that cannot eat lactose, they produce a basic byproduct through fermentation of proteins. These guys don't uptake any dyes at all. Their cell walls become hard because of that basic byproduct, that basic waste, and that basic waste prevents any dyes from being uptaked by their cells. So they remain clear. You cannot see any color change at all in this bacteria. It's just growing on top of the plate. So this is EMB agar. Very interesting little agar that's used specifically for gram negatives and almost exclusively to identify E. coli. And our